Okay, you guys, this is our Zoom office hour, and I'm just doing a video so we could go over differential plates. That's exercise 18 in the lab book. But in the textbook, what will really help is looking at chapter 3 in the textbook. So this is exercise 18 in the differential plates. You will do the last sheet on that page, where you turn in your assignments, attach it to this formal report, which you will write up on grid paper, lab paper, and you will read and look at chapter 3 in the textbook, because it will help with this. Otherwise, just looking at the exercise is not going to be very helpful. That's why I'm going to do a demonstration here. So behind me here we have the 7 media that we're going to be using to inoculate. And then we have these 10 bacteria. All of these bacteria are on your syllabus in the lab syllabus. So if you need to spell anything, I spelled them all out here. But if you have some problem with it, they're all written throughout your lab syllabus. Here, we've used all of these media before at one time, so you're familiar with them. So here's blood agar. This is blood agar plate, BAP. Then we have mannitol salt agar, MSA. McConkey agar, the MAC plate. Eosin methylene blue, EMB. Tryptic soy agar, that's our TSA plate. Sulfur endomotility, that's the SIM tube. This is a tube, by the way. This is a tube. And this is a tube as well. Semicitrate tubes. So, it already says tube. So these are slanted agar tubes. These are the 7 different media that we will be inoculating. In other words, we'll be putting bacteria on these different media. Now the bacteria we're going to be using are these. You're familiar with all these bacteria already. Staphylococcus aureus, SA, Escherichia coli, EC, Bacillus megaterium, BM, Serratia marcescens, SM, Mycobacterium spegmatis, MS, Klebsiella pneumoniae, KP, Proteus vulgaris, PV, Micrococcus luteus, that's Micrococcus luteum or luteus apparently, ML, Pseudomonas aeruginosa, PA, and then the one we didn't use that much, but you do remember that it has this purple or violet color, hence the name Chromobacterium violaceum, CV. These are the 10 bacteria that we will use to inoculate all of these different agars. It's 10 bacteria, and there's going to be too many bacteria to put on one plate. So we're going to use 2 plates per agar. So 2 plates of blood agar, 2 plates of MSA, 2 plates of MAC, 2 plates of EMB, 2 plates of TSA. Why 2 plates? Because if we try to inoculate one petri dish, one plate, with all of this bacteria, it would contaminate. It will overgrow and contaminate the other wells. So we're going to use 2 plates and put 5 bacteria on each plate. All right? So I will demonstrate that. I'll draw it on the board so that you can see what I'm talking about, but just keep that method in mind. Another thing is you can always utilize YouTube videos that go through and explain these methodologies, but I really want to give you a good, solid, concise video here so that you will know what's expected from you on the formal report. Make sure all of this is written on your formal report. That is, of course, on grid paper that you will submit. I think I'll make a place where it says assignments. You will submit this in a place where you'll look in the computer and you'll see assignments. Please submit this in assignments when it's due. Keep all of this information for your unknown. The better and most work that you do here on this assignment, differential plates, this will make your unknown easy, simple, lickety-split. Because this is all the information you'll know. All these bacteria you will see, and all this media you will see for your unknown. All right? So let me just go over differential media and selective media, because that's what you need to know about. Differential media grows several types of microorganisms. It grows a lot of different microorganisms, a lot of bacteria. But they are designed, the differential media is designed to bring out visible differences. So differential media, you will see the difference in all the bacteria that it can grow. Variations, you'll have colony size, the color of the bacteria, and sometimes even the media changes colors. And these are things that you're going to have to check. Did the bacteria change color? Did the media change color? Did they both change color? Sometimes this media could change colors. Sometimes this bacteria could change colors. Sometimes they both can change colors. That's the kind of detail I want to see written up in this paper. Other things like you had a formation of gas bubbles or even precipitation, little things crystallizing out of it. Those are properties and observations I need you to point out in your writing. Differential media contains substances that cause some bacteria to take on an appearance that distinguishes them from another bacteria. That's why it's differential. You can tell the difference. This person or this bacteria turns red. This bacteria turns metallic green. That's a difference. You can look at that bacteria and tell the difference. Staph aureus grows on MSA. Staph aureus grows on MSA. It ferments mannitol, hence the name mannitol salt agar. Plus the fact Staph aureus really likes a high salt, can grow on a high salt content, as well as fermenting this sugar called mannitol. It changes the pH indicator that is in this agar from red to yellow around the colonies. So what changes? The agar changes colors from red to yellow. One important thing to know is that these are really all kind of reddish color. That's why I remember before telling you, don't just go over there and grab the plates, because when you get back to your desk, you won't know what you have. They're all red, different variations of red, but they're all red. The McConkey is a kind of a lighter red, kind of pinkish, but it can still make you think it's maybe mannitol salt, or this one's kind of darker, but anyway, they're all variations of red, so you don't want to get them mixed up. Make sure you look at the colors of the agar in the videos. Just go on YouTube. I'm not going to choose one for you to look at. I sent one to you, but now you guys were telling me that you needed some kind of passcode, which I don't have access to that. But I will check on it. I thought I was doing you guys a favor with sending that URL, but apparently it backfired. So just go on YouTube and feel free to look up each of these agars and see what they look like and what they grow, because that is the kind of information you're going to have to put in a table for me. All right, so with that said, let's talk about selective. So selective media is a little different than differential. Selective media contains one or more agents that inhibits the growth of certain microbes. So certain bacteria won't grow because it's selective. This media may be selective for only a certain kind of bacteria. So mannitol salt agar is definitely selective for Staph aureus. I just said it was differential too, didn't I? Well, perhaps, and you have to let me know, that mannitol salt agar can be a differential and a selective media, can be both. So this certain media will allow certain bacteria to grow on it and inhibit other bacteria from growing. Selective media are very important in the primary isolation of a specific type of microorganism from samples containing mixtures of different species. So maybe you have a mixture of different species and you only want that specific species. Selective is the one to do. Staph aureus will grow very easily in mannitol salt agar, but Staph epidermidis will not. You see, the species are different. One is aureus, one is epidermidis. So that will be a difference. Both genuses are the same. Both are Staphylococcus, but the species were different. And this is where you could put a lot of bacteria in a container and start plating them. And a selective bacteria will show you even the difference in the species. Sometimes antibiotics, dyes, and various inhibitory compounds are often incorporated into the media to create selective conditions for growing specific organisms. When we say sometimes dyes are put in the media to make sure that only certain organisms grow, we'll see that example with eosin methylene blue. You guys are familiar with methylene blue as a dye. This was put in this medium to make it selective. This type of medium is selective for only gram negatives, right? Only gram negatives. And usually in the ones that ferment lactose. When they ferment lactose here in the gram negatives, they give a kind of a greenish sheen on the agar. McConkey is also selective in that it only allows gram negatives to grow. It inhibits gram positives, just like EMB. So McConkey and EMB are both selective, and they will inhibit gram positives, but they love to grow gram negatives. EMB will grow lactose fermenters, and you will see like a really nice greenish sheen on them. This information is what I want to see in your table when you talk about each of the media and what changes you saw. When you do blood agar, for example, I want you to look for hemolysis. That's where the blood has been cleared away. Blood hemolysis means the blood is lysed. We talked about that a bit before. If you need to see that, turn to look in chapter 3 of your text, and it will give you different examples of that. We talked about hemolysis when we did streptococcus. You had alpha hemolysis, beta hemolysis, remember, and I think a gamma hemolysis. It's the way the blood clears from around that particular bacteria. And TSA, just about everything grows with TSA. So this is kind of a multi-cultural type of agar that really most organisms will grow on it, as well as blood agar plate as well. Remember for sulfur endomotility, the SIM tube, please remember to use a needle for this one to stab it into the tube and then bring it straight out. And for this citrate, use an agar slant, so you will start at the bottom and wiggle your way up. So we've talked about all of the media. It's up to you to go over all this bacteria so you'll know which one is gram positive, which one is gram negative. I want that in your report as well, so I will know that you know what you're talking about. Because when you get ready to do your unknown, the first thing you're going to do is your gram stain. Okay, next we'll talk about broth cultures that you're going to use to inoculate all of your plates. Okay, let's go on and continue with our Zoom office hours, and we're just going over the differential plates, exercise 18, because that exercise does not explain anything to do with this experiment. Please remember to do the lab exercise at the end of that chapter, at the end of that exercise, and attach it to your formal report that you're turning in for this. All right, let's look at what we've drawn up here. So, of course, you're going to use aseptic technique, always, you know, flame the mouth of the tube. When you open it, flame the mouth of the tube. When you put the cap back on, flame your loop, flame your needle, just flame everything. And let it cool so you don't kill the bacteria, because you want to have something to grow. So using aseptic technique, you're going to transfer these broth cultures. All these are in broth. You see that they're turbid. Turbidity means what? That there's growth. It's cloudy, there's growth in here, right? All of these 10 bacteria have growth in them. You're going to transfer these 10 bacteria into sterile cultures, all right? Into sterile broth, onto sterile agar. All right, so these drawn up here, these are all sterile. You notice there's no turbidity, they're all clear. So after you inoculate them, we're hoping that you get some growth where it should be. Here I've drawn a needle, and here is a loop. Flame your loop, flame your needle, flame the mouth of your tube. You'll remember that as being aseptic technique. Now, if you recall, all of these are different. This is Staph aureus, Escherichia coli, Bacillus magnetarium, Serratia marcescens, Mycobacterium spagnatus, Klebsiella pneumoniae, Proteus vulgaris, Micrococcus luteus, Pseudomonas aeruginosa, and Chromiobacterium violaceum. And these are all inoculated, been waiting for you to come and get some of it out and start to inoculate your sterile media here. So how are we going to take 10 bacteria and put them into all of these? That's a good question. The first one, the first plate that we will use will be the blood agar. So I've drawn it like this so I can put BAP here. And then it's 2 blood agar plates, because 10 bacteria on one plate is going to overgrow the plate. Everything will be contaminated. So I got 2 plates, and I'm going to divide it into 5. 5 bacteria will go to this blood agar plate. 5 bacteria will go on this blood agar plate. Then that means all 10 bacteria will have been placed on the plates. So just draw some spokes to a wheel like this to make 5 places. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Everybody, I want you to go in this specific order. So every paper will have the same order. So when I'm checking it, I don't have to look all over for everything. Keep everything orderly. And make sure you use ink. You know that. So we'll start with this blood agar plate. Another thing, remember, your name and the date goes on each plate. Your name and the date goes on each plate. And you need to know what kind of plate it is. So we already labeled it as the blood agar plate. So make sure you label your plates properly. Let's go on and put the bacteria name in each well, so that when we get ready to inoculate, we can put the correct bacteria in the correct well, okay? So this, and I want everybody's to be the same. So please follow this example. So that's SA, MS, SM, EC, and 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Oh, okay. This should be SA, MS, SM, so that's correct. Got a little off. This is Yeah, I'm gonna just make this the way this is, like this. And this should be BM and then E. coli. So I want it in that in that arrangement, so when I'm looking at it, I can see it really quickly. This one will be KP, CV, PA, ML, and PV. So now we have all the bacteria that we're going to inoculate in these sterile wells. We have it all labeled, so we'll know that SA goes here, EC goes here, BM goes here, SM will be here, MS will be here. So make sure it's in that order, please. This next one, let's do this as MSA. So just so I know which agar this is, because remember, they're all kind of red, so they all look alike. And I mean that in the best way, it does all look alike. So here is, let's make these, I don't know why, 5 wells, right? And then this one. Yeah. So this one's the same. It would be SA here, just like the SA is over here. So what goes here? MS, correct? SM. Same thing as this one. BM, and then EC. This is a different agar though. This is MSA. It's not BA. It's not the blood agar. This one will match this one. So we see the KP is here, the CV, the PA, the ML, and the PV is here. Make sure your name and the date goes on each plate. I'm not going to stress that enough. I need to see that you do everything properly. Everything must be done properly. And if you don't know or you don't remember, look it up. No laziness on this paper. It's a formal report, and I'm going to be checking it because you got a lot more time to do this thing now. So I'm going to be checking it with a fine-tooth comb. So that's the MSA label. I'm just going to do all of them. Let's do the MAC plate next. The MAC. Let's get these 10 bacteria on here. All right, so we know the order. SA here, MS here, SM, BM, EC. This one is KP, CV, let's see, what's this, PA, ML, and PV. All right, we got the MAC plate all labeled up. Let's go over to the EMB plate. This is EMB. EMB agar, and we have to load it up with the SA, MS, SM, BM, and the EC, same. And then this one, KP here, CV, PA, ML, PV. So all of them are labeled exactly the same, but you know that these are all 5 different agars, right? And then let's do the last one. This is a TSA. Just about everything goes on TSA, but we must label it so you'll know which plate this is. And let's just go on and make our wells here. Oops. All right, let's label it SA, MS, SM. Keep it just like this. And EC, I want it all organized exactly the way it is on this board. I mean, as far as the bacteria is labeled. I don't want it all on one page like this. This is too messy. Please use different pages and spread it out. Don't cramp it up like this. But this is just so I can get the video in and get the gist of it. But the point is, I want the bacteria labeled in each well exactly the same as this on each of the augers, okay? So that is that. Now what's the next thing that we do? Let's go on and just label all of this stuff so we'll know what next. So here we are with and I'm going in the same order as the bacteria that are already inoculated, the inoculated broths here. So let's look at the semicitrate. So we will label this maybe with a different color just for fun. We'll say this is the SA, the EC, the BM, the SM, MS, KP, PV, ML, PA, and then the CV. Make sure your name and the date go on here, on all your tubes. Okay, just so that I will know that you know to do that. This one here, this is the Sims tubes with the needle. I showed here that when you poke the needle down in here, you drag the needle straight out, and then in about 24 to 48 hours, you should see growth if it is one that will grow in this type of medium. But that one I want you to remember is the Sim tube, you use a needle. Let's go on and label them the same as this. SA, EC, BM, SM, MS, KP, PV, ML, PACV, right? And that should be really great with your labeling. Spread these things out on sheets of paper. You got a lot of paper, so just use it because we haven't been using it in the lab, so you might as well just go on and use it. And please note that for the semicitrate, you're going to use a loop, and for the Sims tube, you're going to use a needle. And let's go and talk about what you're going to do with that. So now you will flame, so let's go and inoculate the plates, okay? These are all the things that you're going to use. These are all your bacteria that you're going to use to inoculate your plates. Where do you think the SA, I flame my loop, flame the mouth of my tube, now I'm ready to put the loop in. Where do you think you're going to inoculate it? Of course, SA will correspond to SA here. So all you'll do is make a little swiggle to show that you inoculated that SA, right? With that, since this is all sterile, anyway, just for technique, you'll flame your loop again, go back in and get some SA, and do another swiggle. Since this is all sterile, really, you could just keep going in the SA and doing all your SA swiggles. You can inoculate all of your SAs, and then you're finished with SA. You might as well just go on, and I would flame my loop though after this, and go back in, flame the mouth, get some SA, and then you might as well just start here. Remember, you start here, and let me move that out of the way, and you swiggle up so that you can just go on and inoculate all your SAs. All of your Staph aureus are now inoculated with Staph aureus. Now the next one is Escherichia coli. Flame your loop, flame the mouth, go in and get your Escherichia coli. Inoculate the EC where it says Escherichia coli here. Inoculate this one, that one, and this one. So you will inoculate all those. And then, flame your loop, flame the mouth, get some more EC like that. And each time though, I would get some to make sure. And start here and inoculate this from the point down here, swiggle up, and now you're done. Make sure you flame the mouth, flame the loop each time. But I would definitely just go in and do all of them that I have that are available to inoculate at that time, as opposed to going back and forth, changing up the tubes. That gets to be pretty tiresome. I'm looking for this one, another color. Let's go into B, Bacillus megaterium. Flame the loop, flame the mouth of the tube. Go in and get your BM. Inoculate all your BMs. Now olive and in between, of course, you're flaming the loop, flaming the mouth, etc. And you might as well just go in and inoculate this. Start here, wiggle up, streak up, and now that's inoculated. You guys get the gist of this? So the point is that you will go into each one of these using aseptic technique. So SM, you will go in and inoculate SM in all of them. Some people do it like this. This is the way I prefer, because then you don't get any really contamination. You just know that loop is specific for that one. And then you just go in and inoculate that one. Go in, do your MS here, and flame everything, and then go and do all your MS. And just say that one, okay? Now we're at the next one, and flame everything. Do your KP on all of them. I think you get the gist of this. Then you might as well. But each time, make sure you're flaming the loop and flaming the mouth, flaming the loop, flaming the mouth, and go in and get it. Let the loop cool, and then inoculate the agar. So we're just going to finish up by getting Proteus vulgaris here, and here, and here, and here, and here, and then there. And Micrococcus luteus here, swiggle. Make sure you don't go past any of the lines. Do not go over into here. Just make sure, because you do not want this stuff to grow over into your other wells. So you know Serratia marcescens is notorious for going over into other wells. Finish up here and here. Pseudomonas cerviginosa. Just do all of those. Oh, I forgot to do ML. Looks like I just did that with the Pseudomonas loop. Don't do that. And then Chromiobacterium violacum. And just finish up. And now, oops, I missed those. So now you have all 10 of your bacteria being inoculated that were once inoculated, all the sterile broths and the sterile tubes, right? Now that you're finished with all the loop work, you could go in and use your needle here, flame the loop, flame the needle, flame the mouth of the tube, flame the mouth of this tube, of course. You have to flame these two, and then just stab it down in, bring it back up, flame the needle, go back, flame the mouth of this Escherichia coli, get some out, put it and flame the mouth of this tube, stab it, and then you just go on. Anyway, you'll end up flaming and stabbing all of these. Make sure when you go in, you bring the needle straight back up so you don't wiggle at all. And that way, you can say you inoculate your sim tube. And if you have growth, it'll look like that. It'll be some growth around the area of where you stab. So that is inoculation in a nutshell. I was trying to do it with all different colors, but ran out of color. But you get the gist of it. The next thing I will talk about is the table of where how I want you to set it up in your data sheet. So the first part of this, when it was just all clear and you just drew all of your bacteria here, that was part of your procedures. Now that you've grown, you inoculated, and say it's 2 days later, like Wednesday, because you're doing this lab today, now you have your bacteria has grown. So now these are results now. These would be considered what you did the day of your procedure. And then now these would be, like, I'm just saying, the next time would be results, okay? So this we'll just say this represents the growth stuff, but I'm just showing you how it should look. Let's go on to the next thing, which will be the table, and how I want you to present this as data in your results. Okay, so here we are now with the table that will go into your results section, your data table results section. Here we have all the media going across this way. That would be the x-axis. You have your blood agar. You could have just put BAP, BAP, the mannitol salt agar, the McConkey agar, the eosin methylene blue, the trypsin soy agar, and the Sims tube, and the citrate tube. Now, for purposes of fitting everything on this board, my little squares are small, but yours need to be larger so that you can put enough information in them. Along this y-axis, we have bacteria. All 10 of the bacteria are labeled here. Staph aureus, E. coli, Bacillus megaterium, Serratia marcescens, Mycobacterium spagmatis, Klebsiella pneumoniae, Proteus vulgaris, Micrococcus notius, Pseudomonas aeruginosa, and Chromiobacterium violaceum. All of those are labeled here. You can number them. That would be fine too. But my space is confined, so I don't want you to have these little narrow windows to write in, because you have to put, for blood agar, does Staph aureus grow on blood agar? So we're going to be looking for growth. You'll put growth, just as an example, you'll put growth, and you can put a positive. Did you see any hemolysis? So with this one, blood agar, you're going to be looking, so we are going to be looking for growth, plus or minus. You're going to be looking for, in the blood agar one anyway, any hemolysis, just for this one mainly. That would be for the BAP plate, which you can name that. You don't have to make blood agar, you put BAP. I just was hurrying up. Color changes. I want you to note, did you have any color changes with the media? Did the media change colors? Or did the bacteria? Or maybe the media and the bacteria change colors. Please indicate that in these squares, which should be a lot larger than this. Please make them larger. Any motility? Let's see, we can go over here. And of course, we're looking for motility here in the Sim plate, tube. So if you see motility, did the color in the Sim citrate color change? And if it did, what color did it change? Don't just say yes, it changed colors. What color did the agar change? What color did the bacteria change? You must indicate that. Okay? This table is going to be the table for your unknown. So you want to put as much information in it concerning each of these bacteria and how it relates to each of these media as possible. Your squares can be a lot larger than this, because this is what you're going to use as your key to determine your unknown. So we'll go along with Staph aureus. Look, you can see a blood agar. Yes, there was growth. No color change, whatever. Oh yeah, we'll just say hemolysis. Yes. And for this one, I think it's a beta hemolysis. You know, you have the alpha, beta, and I think a gamma. You could put which hemolysis it is. You are going to have to do some investigation. You're going to have to do some research. And that way I'll know that you understand what each of these media are doing and how each of the bacteria respond in these different media. Now, mannitol salt agar, you know that Staph aureus, this is selective for, this media is selective for Staph aureus. So that is definitely some growth. The media will change, and we mentioned that before. The media changed from red, remember that's red, to a yellowish color. You must indicate that. You must indicate that. So we're going to look at Staph aureus here as it goes with MAC. Does MAC like the Staph aureus? Would it prefer to grow on MAC? No. So it's going to be no growth, because MAC as well as EMB grow gram-positives. Now EMB can also be around with lactose fermenters. Remember, they leave like a greenish sheen on lactose fermenters, but all of these are enteric bacteria. Remember when we talked about enterobacteriaceae? All these are things that come from the gut. So when we look at that one, we will look at E. coli and know that it is a gram-negative. That's why you have to put, if these bacteria are gram-negatives or gram-positives, you could put them right here, gram-negative, gram-positive. Like you could just go through and put a positive, a negative, and put gram here, so that I'll know that that's what that is, gram-positive, and then going down, so I'll know. That way you will remember that only gram-negatives can grow in MAC and EMB. So E. coli will grow here and here, but not where it's selective for MSA, for staph. And then you look at blood agar, tell me if it's hemolyzed or hemolyzed or did it grow or not. TSA, let's finish up for Staph aureus. Everything just about grows on TSA, so that would be a positive. SIMS, Staph aureus really doesn't have motility, so we'll just say that's a negative. Semicitrate, you'll have to check the color for this one. You can see, tell me if something grew on the top of the slant, because you can see some kind of creamy looking kind of whitish film, and you may have growth, but it may not have changed the color of the semicitrate too. So you'll have to tell me that once you investigate what the semicitrate is. You know, just let me know, does that bacteria utilize citrate? Was there any growth there on top of that slant? You'll go through each of those, and like for E. coli, you look at blood agar, you look at MSA, MAC, then you'll do BM, look at this one, look at it in each one, and then write down something. Make sure that your space is much larger than this one, because I just did it for space sake. But this is the way your table will be set up. I want it exactly in this order as far as the bacteria lined up here. I don't want it random, because I don't want to have to look at 18 different pieces of paper that's all over the place. And it's like we both have enough to do, so let's make our lives a little easier. And then the media should be also in this particular order. Put as much information in this table, in this chart, as possible. So your investigation is what's going to determine how easy your unknown is going to be. Because once you get your unknown, and I give you a little bit of information about it, you're going to tell me the steps, what you went through, and then you'll be able to use this chart as your kind of a sheet that is going to give you the information that you need. This will go under assignments. I will make a new little space where assignments can be turned in for your formal reports. We'll go under assignments. Please remember that we're going to incubate the test tubes and the at 37 degrees for about 24 to 48 hours. Sometimes in our book it says 18 to 48 hours, whatever. Just incubate it long enough so we get some growth. Remember to invert them, to make them upside down so no condensation occurs on the bacteria on the media itself. And I do want to remind you that for Zoom now, for some reason, I don't know why, but this is a password that we're going to need. I think they're just overwhelmed and they're just trying to make it a little more difficult, especially since we're using the free one. So remember that the password, I'm just going to put it right here, is 6, capital F, lowercase f, J, lowercase j, and lowercase I. 6, capital F, lowercase f, lowercase j, lowercase I. That's the password for Zoom now. Hope you see that. And remember that when you see this video, email me so that I will know that that was your attendance, because I know we had a problem with our Zoom because we needed this password for some crazy reason. Look at YouTube videos. I tried to go through some YouTubes and I sent a couple of links, but people are telling me now that they need another code or password. So many passwords, so many codes, because everybody's online and people are infiltrating different media, not this kind of media, but the different types of ways to contact and inform people. And it's getting, I would say, it's getting contaminated. So that's why they have different passwords. So like I said, you can email your questions then at our next Zoom meeting. Please remember, this is the password to get into Zoom now. And we will start with viruses next time. That video will be up and coming soon. In the meantime, make sure you go over exercise 18. Be sure to include the last page of exercise 18 with the work that you're turning in, and check and read the textbook chapter 3 where it says selective and differential media. Just flip through it, it'll give you some great examples. Feel free to look at YouTube videos. They have them with each of these augers, and I mean, it will explain what grows on it, what doesn't, and what to look for. That is the kind of information I want you to infiltrate into your test, into your data sheet, and put it in there. Make sure you put your discussion, and I want you to discuss each of these media as it relates to each of these bacteria. In other words, you're going to discuss this table. That's what you do in discussion. You discuss the results. And then I want a conclusion. So make it worthy of a college lab assignment, and I will see you next time when we start with the lecture series. This is just for the lab so that you can understand it and do an excellent job on your formal report. I look forward to reading it.